All right, so we're back for the last half an hour. So let's continue. Right, so we're talking about now genetics that have gone beyond what we know from our friend there, right? So here are some of the things that we're going to be covering. Mendel never knew about these simply because the Mendelian experiments were done primarily on, of course, as you know, peas. Peas, for the most part, are dominant recessive, which is why all of his conclusions would have been based on that model of things, right? Now, we're going to have to learn about incomplete dominance, codominance, as well as multiple alleles. Remember now, these are not Mendelian. So we can call them non-Mendelian genetics, okay? So all six of these are non-Mendelian genetics. So let's start with incomplete dominance. In incomplete dominance, you do not have, once again, dominant and recessive, okay? Instead, you have a blending occurring, right? It's also different from the codominance we mentioned earlier. We have a blending effect occurring. So the idea is that in the case of incomplete dominance, both alleles want to show their traits, right? So let's say, for example, we have a plant with incomplete dominance because it shows up in plants quite a bit. We have one red plant with red flowers, I should say, and one with white flowers. Well, here we go, right? Now, with red and white flowers, both genes for red and white want to show up, right? What we have here is the parent generation, right? The parent generation is going to, of course, be homozygous dominant, right? So over this side now, we're going to have two genes for being red, okay? But because this is not dominant and recessive, we cannot have small r over here. Instead, we have to pick another letter to represent this. And of course, you notice I'm writing my letters as capital, right? So we have another one for white. So for example, I could just do doubly for white. So this would be our parent generation. Homozygous dominant, homozygous dominant, homozygous dominant, okay? But two separate traits. Now, the thing with incomplete dominance is that, as I said to you, both of them are trying to make their trait come about at the same time. So that means if you have the gene for red flowers, it means that you have the genes for the making of the compounds, right? So in the flower, you're making compounds that will appear to be red, giving the flower a red color. If you have another gene for white flowers, then now that gene also will result in the production of the compound that gives you white flowers. So now we have one red flower compound and one white flower compound all in the same flower. When they mix together, of course, as you know, red plus white gives you pink. Pink is basically pale red, right? As a matter of fact, I don't even know this, but there did not used to be a word for pink. The actual term pink popped up in the 1800s. It was basically dark red, light red, right? There was no pink. So pink that we know today used to just be light red. But anyway, we call this pink now, right? Now, pink, of course, it means you have one gene for the red color, one gene for the white color. Both of them are trying to be seen, hence you compromise at pink, right? Now, if you cross now the F1 generation, so parental generation, homozygous recessive, the F1 generation now, that's first filial generation, are going to, of course, be heterozygous. Both of them, which is why they are pink. So when you cross heterozygous now, normally remember when you cross heterozygous for the monohybrid cross, you would end up with a 1:2:1 genotypic ratio and a 3:1 phenotypic ratio, right? But here, so we have a 1:2:1 ratio, right? So we have for red, 2 R's, for pink, each of them has an R and a W. And for white, 2 W's. Which of course means that we have a genotypic ratio, 1:2:1, and a phenotypic ratio, also 1:2:1. Okay? So in the case of incomplete dominance, the genotypic ratio and the phenotypic ratio are going to be the same. Okay? All right. So far, so good, hopefully, right? So, of course, here is a Punnett square, which is basically showing the same thing, right? I think they used R comma for white for them and just capital R for red, okay? Which, I mean, that works too, right? But normally I just pick a different letter. It makes it less confusing. But they get the same results, right? So they get 1:2:1 ratio, same way, okay? So hopefully that makes sense, right? So if you cross two plants and the offspring have phenotype that neither parent has, or neither that did not appear in the previous parental generation, chances are you're gonna have incomplete dominance, okay? So once you have a blending of phenotypes in the next generation, that is going to be incomplete dominance, okay? So a blending equals incomplete dominance. So you don't get red or white, you get a third option, which is pink. Now, codominance is a bit different. Remember, we spoke about codominance when I was talking about sickle cell anemia. In codominance, you're not blending the phenotypes together to get something different. Instead, in codominance, both phenotypes have to be expressed in their natural way. Okay? So both phenotypes have to pop up in a very obvious way. So here we go now. We have here alleles for red and white hair on cattle. Okay? So we have capital R's representing red hair on cows, and we have capital W's representing white hair. Very similar to what we do with the flowers, right? Now, if you have cattle with both alleles, they tend to have a pattern that looks brown. That says brown and white patterning, or roan, right? So you cross red and white, it gives you kind of a brownish color, apparently, right? It also mentions that in certain varieties of chicken, you also get allele for black and white feathers are being codominant, right? So you have this spotted chicken look. I don't really see a lot of spotted chickens running around Jamaica, but I've seen pictures of them on the internet, right? So this here now is a roan cow. Here's the idea. We have white patches here obvious white patches, right? But what about this kind of brownish color? You basically get a mixture of red and white hairs, and it makes it look brown. So the white hairs in there sort of diffuse the color, right? So we have our essentially we still have our red, and we also have our white. So both phenotypes have shown up in this animal, okay? Otherwise, red and white should give you pink, right? I've never seen a pink cow in my life, so I don't think it works that way, right? So, incomplete dominance versus codominance. Remember, in incomplete dominance, you blend the two traits together to form a third trait. Red, white equals pink, right? In codominance, on the other hand, both traits have to show up. So red, white equals red and white patches, right?
So can I ask a question now?
Of course you can.
Okay, so incomplete dominance is blending.
Right.
So black and white, gray,
Mm-hmm.
red and white, pink. But what if it's like, what if it's black and white and then you get brown? Is it the same? Is it still incomplete dominance?
Well, it depends. Remember, sometimes the fur of an animal looks black because it is very concentrated melanin. So extremely dark brown looks black to us, right? And if so be the case, if you get brown when you mix a black and a white together, it means that you have diluted the melanin with the white, which is why now, since there's less melanin, it now looks white. I'm sorry, brown. So if black and white give you brown, that is also incomplete dominance, right? So
Okay,
here we
so
have roan cattle again. Once again, you can see red patches, you can see white patches. Hence, this is incomplete dominance. You can see both the traits showing up, right? So incomplete. Right now, I give you a pick. If it was codominance, red and white would give you a flowers with red red and white patches on it. I don't know if you've ever seen like flowers with multiple colors on the same flower. That is codominance, right? So yeah, actually, I have seen this
Can,
out here. Yeah,
can incomplete dominance happen with other traits besides color?
like what? Give me an example here, me. I mean, as long as you can
Um,
think of an intermediate phase between the two traits, absolutely. I mean,
okay.
height is not really something you can consider for that, but other things. For example, think of human skin color, right? Think of the range of skin colors that, well, I mean, skin colors are another one that's a bit more complicated than that. Um, yeah. But yeah,
So does incomplete dominance play a role like in genetic disorders? You think?
um, generally speaking, incomplete dominance is supposed to give you what amounts to a third trait, right? I know codominance does play a role, but I can't off the top of my head think of any genetic disorders where incomplete dominance is playing a direct role. Doesn't mean they aren't there, just means I can't think of any right now. So if they are there, they're not very common. But I look into it though, because that does sound interesting. Now, this kind of chicken I've seen in people's backyards. I don't really see people growing them commercially, really. This here is codominance because you have a white chicken and a black chicken gives you can clearly see you have black and white feathers in here, right? So that is codominance. If the feathers were all gray, then that would be incomplete dominance. All right. Then we have multiple alleles. Now, this is very, very important with regards to things like blood type, okay? your blood type is multiple alleles. What I mean by that is this. Okay. Any of you know your blood type by chance?
Yes, sir.
What's your blood type?
A positive.
That's the second most common blood type in Jamaica. Anybody else know the blood type?
AB.
B negative.
AB what? Did they tell you AB what or just AB? Because that's a bit on the vague side. I think somebody said somebody had a negative blood type.
B negative.
B negative. B negative is what? I think is the third least common blood type in Jamaica. So it's a relatively rare blood type, right? A negative being the rarest blood type in Jamaica. Now, here is how that works. We have 3 alleles for blood type. So we have type A allele, B allele, and we also have one for D. D is the rhesus factor. That's what makes your blood positive or negative. Okay. We also have a recessive allele called O. Okay. So these 3 here are dominant. Well, technically, these 2 here are codominant. This one here is just regular dominant, and this one here is recessive. Right? Because of that, if you are somebody with blood type A, okay, let's leave out the positive for now. So the D makes it blood positive or negative, but let's leave it out for now. So if your blood type is A, you can have 2 alleles for A like this. And you said, I remember I said to you A and B are codominant, right? Which is why I write it like this. I is for immunoglobulin. Sorry. And then A, of course, is a blood type. You can also be blood type A like this. This is how they write O, right? You notice that this is a common I. That's to show it's recessive. If your blood type is B, I think somebody said the blood type was B. Remember, I'm leaving off the rhesus factor. Then your blood type can be like this, which is probably unlikely. More likely than not, this is your blood type. Right? The reason I said that this is the most likely blood type is because the recessive allele is more common than the B allele by quite a bit. Right? It's more than twice as common. So chances are, if your blood type is B, you're more than likely going to be heterozygous for B like this. Okay? Now, because of this setup, let's get rid of this. Because of this codominant setup, it means that if you have 2 people, let's say you have a person with type heterozygous blood type A, heterozygous blood type B, and they have a child. Now, once again, I'm leaving out the rhesus factor, right? Blood, about, there you go, 23% of Jamaicans are blood type A, and 20% are blood type B, right? So it is not out of the realm of possibility that these 2 people will meet. It probably happens every day. So let us say now we have this. So blood type A, and then we have blood type B, having a child. Now, obviously, this one here can contribute either the A allele or the O allele. right? Remember I write O like this because it is recessive, right? So capital I is a dominant, common I is recessive. Of course, the B person can donate that, okay? So if we throw this in a Punnett square now, we of course end up with this here. Right? So we have one AB. So remember, being codominant, this person's blood will show the traits for both A and B blood. Then we have one type A, just normal, which is heterozygous for A. We have one type B, that is heterozygous for B, just like so those are just like the parents. And of course, there's a chance to have a child with blood type O. Okay? So notice here, there ain't no 1 to 2 to 1 ratio here. Our ratio is going to be equal to 1 AB, 1 A, 1 B, and 1 O. So we have a ratio of 1 to 1 to 1, right? So there's a 25% chance of any of these blood types coming about from these two parents. So 2 parents, 4 different blood types, right? Now, the multiple allele thing is one of the reasons why blood types cannot take the place of a DNA test when determining paternity. Y'all know paternity test, right? But does that make sense to you? Does it make sense to you why you can't use a blood test to determine paternity? I just showed you that there's a genetic relationship between the parent's blood type and the offspring's blood type. I then said to you, you can't use that to determine who the daddy is. Do you all see why that is? If not, let me know because that's a question I can come again. Does it make sense to you? Or you can't use a blood test for this? All right, I guess you're all good. Nobody's saying me. Let's continue.
No, sir. No, sir. Let me just say that then.
Okay.
So you know, everybody can be sure, and even myself too as well.
All right. Here's the thing. If you are blood type, let us say you have a child whose blood type is O. Okay? Let's use this scenario. The daddy is blood type B, the mommy is blood type A, the child comes out O. I can say, hey, guess what? You're heterozygous for A, he's heterozygous for B, the baby is yours. Right? But guess what? If the other, all it is saying to me is any blood type containing a recessive gene could be the daddy. So it could be another blood type A up here, so. So IA with I, and it pops out as blood type O, right? Not to mention the fact that anybody with this blood type could potentially be the father, right? So all the blood types do is they group people into big categories and say, guess what? Anybody with these genetic characteristics could potentially be the father of this baby. How do I know it's you specifically? I don't know, but it's possible. So therefore, you're the daddy. Right? It's the equivalent of the judge saying somebody was murdered, you are nearby, therefore you killed him. Right? It is not anywhere near accurate enough to be a paternity test. But we used to use it to determine paternity. And would it surprise you to know that in Jamaica, right? 30% of paternity tests done come back negative for the father. So 1 in 3 people who do a paternity test, as normally the males ask for it, is not their pitney. Regardless of what the blood type is, it come back as not being their children, right? So therefore, having a blood type that could potentially come from somebody else, if they were your father, does not mean they're your father. It just means this group of people with this blood type, any man in that group could be your father. Does that make sense, everybody?
Sir, I get your explanation, but how would you structure your answer? Like, how would you word it, like on
I was hoping,
an
all right.
exam?
You would indicate that blood types do not represent specific genetic combinations, except for with regards to the blood type itself. Therefore, people of very little or limited genetic similarity can be similar specifically for genes associated with blood type, and will therefore pass on their blood type genes to their offspring, just like everybody else with those specific genetic traits. But this does not preclude someone not being directly related to a person having these traits, right? In other words, having the same genes for blood does not equate to being the father of an individual. It's like saying everybody with genes for dark skin are directly related to one another, or everybody with blue eyes are related to one another. Everybody with brown hair related to one another. That is not the way genetics works. Okay, at least not directly. So therefore, DNA tests which look at thousands of genes and compare those thousands of genes to determine the likelihood of two people being related are far more accurate than a blood test that is only looking at a handful of genes to determine the same thing. All right? Hopefully that makes sense, right? So yes, a blood test is based on genetics, but the number of genes that they look at are way too narrow a scope of genes, and those genes are way too common to be used for any sort of test of relationships, right? All right. So that's blood. They give you an example now in rabbit coat color, right? It says the rabbit coat color is determined by a single gene with 4 alleles. We have type C, chinchilla. That chinchilla was an animal, not a color. Well, no, chinchillas are animals. What the hell is this? All right, whatever. Himalayan. Okay, how is Himalayan a color? Seriously. Himalayan is a mountain, isn't it? An albino. I'm going to say something very sexist. I'm sure as a woman thought of chinchilla and Himalayan as colors, right? Men, what are colors? Red, green, and blue. Women, peach, chartreuse. Don't make things too complicated. So we have a red rabbit, a gray rabbit with varying colors of gray, and then a black and white patched rabbit. So Himalayan is black and white patched. Chinchilla is varying color, gray intensities, and of course, albino is white. Okay. So it says here the genes that can cause this to come about. So we have the normal wild type gene, which is C, which looks like it gives like a reddish color. Then we have the chinchilla hybrid of that gene, not hybrid, the chinchilla variant of that gene, which is CCH. And then we have the Himalayan, which is CH, and the albino, which is small c. So albino looks like it is recessive. These two would have to be codominant because if you notice here, they have a superscript, right? So let's see how this goes. All right. Can anybody tell me what is this one here? Is it chinchilla? Is it Himalayan? Is it what is this? Somebody help me out. What is this red?
I would say Himalayan.
Himalayan, sir.
All right, I hear 2 Himalayans. Himalayan is supposed to be red and oh sorry, black and white. Okay. And this one here, is this one red? It looks red to me. Kind of brownish red.
Yeah, wild type.
So we'll assume this is the wild type. All right, good. So we have hopefully what amounts to a Himalayan here and a wild type. All right. Now, if you cross these, well, you can't get any of the other ones from it, can you? If you cross a chinchilla and a Himalayan, what are you going to get? I have no idea. But here's blood again, which is easier to understand, right? So you have type O blood, once again, 2 recessive genes, type AB blood, etc., which we spoke about earlier. But I will mention, we did this in the AMP1 already. So y'all remember that the importance of the genetics of blood comes from when you're trying to give blood, right? Somebody with the wrong blood antigens cannot receive blood from you. So somebody with type A antigens cannot receive blood from somebody with type B antigens. That will, of course, cause a serious problem, right? And type B can't take type A, etc., right? So hopefully I'll remember that from NP1. Now, the last one, and then we call it a night. Then we have polygenetic traits. Now, of course, as you know, poly means many, multiples. And genic, of course, is going to be genetics. So polygenetic traits now is looking at a trait that is controlled by multiple genes. You can think of each gene adds something to the trait to strengthen the trait, right? So it mentions here the length of stems in plants, all right, or eye color in fruit flies, controlled by 3 different genes, and human skin color is controlled by more than 4 genes, right? Let me just briefly mention human skin. Well, let me see if it's mentioned here. Yeah, here we go. Let's just do human skin color first, because the rest of this is kind of boring. Human skin color. Here's the basic idea. You have 4 genes of skin color, as I mentioned. A, B, C. Well, that's 3. 4 different genes. All right. Somebody can't count because I'm seeing A, B, and C here. Where they get 4 from that? Anyway, we'll work with what we have. So we have gene A, which can be dominant or recessive. We have gene B, which can be dominant or recessive, and gene C, which can also be dominant or recessive, okay? The idea here is this. The dominant genes make your skin darker, right? Dominant genes make your skin darker. Recessive genes make your skin lighter. Now, depending upon the combination of dominant genes and recessive genes, that is what determines your actual skin color. So, for example, here on the light end of the spectrum, out of 64 combinations, this person here has one dark gene. Okay? So 1 out of 64 dark genes. Therefore, remember, the more recessive genes you have, the lighter your skin is. So this person here has very light skin because it only has 1 dominant gene. 6 out of 64, you have tanned skin. So you're still fairly light, but you get a little color in there. It's kind of like a Mediterranean color. So people from like Greece and Italy, that's the kind of skin color we're talking about. Still look like white people, just white people with a tan. Then now things start to get complicated once you hit the next combo here, right? So we can see here, out of the 6 genes here present, this one has 1 here, dominant gene. So that's 6 out of 64, that's 10%. So 10% of the genes here are dominant, which gives you the tan. Here now, we have 15 out of 64. So more than 20% of the genes are dominant. Now, this is the skin color. I guess we'd say browning, in Jamaican parlance, right? That said, there's going to be some overlap between these two, but still. Then now 20 out of 64, you're still on the lighter skin end of things, right? So I say you're basically half and half. Then now, once you have 4 genes for skin color, your skin is starting to look more obviously darker. You're not dark yet, right? But your skin is more brown than it is light. Now, most Jamaicans are going to be somewhere between these two. Okay? So most Jamaicans will have either 4 or 5 of these dominant genes for skin color. It's very rare to find somebody with complete dominant genes for skin color. You ever hear people say, oh, somebody black till them shine? That's the kind of thing we're talking about, right? You really don't see it very often. Even among so-called people of African descent, you don't really see skin color this dark normally. So even in Africa, there's like, yo, that brother, the blackie man, right? So yeah. Remember, the idea here is that you have these genes, right? 6 of them, they can be dominant or recessive. The more dominant genes you have, the darker your skin becomes. So each gene adds something, adds an increase in the production of melanin that will make your skin light, darker and darker and darker, right? Now, I want to point out that the genes for skin color are not related to the genes for facial features, for example. So it is possible for somebody to look very Indian and be extremely dark skinned, right? Or somebody to have very, let's call it sub-Saharan features, broad nose, round face, that kind of thing, but have extremely light skin color, right? So don't equate facial features with skin color. It just so happens that sub-Saharan Africans are more likely to have more dark genes in them. That's how we normally think about. When we think of black people, it's normally sub-Saharan Africans we think of, right? Whereas the lighter skinned Africans tend to be North Africans, as in above the Sahara Desert, right? So yeah. All right. Hopefully that makes sense to you, right? I'll probably go back to it again in the next class. But for now, we would have stopped at polygenetic traits, okay? All right, it's 8 o'clock. So I'll stop here. And I will speak to you all, I think, tomorrow. It's tomorrow. One second. I think we have class tomorrow, is it? All right, hold on. Let me bring up my time. I honestly don't remember what time I have class tomorrow. Tomorrow at 2 o'clock. Okay, yes. So I'll speak to you all tomorrow at 2 o'clock, 2 to 4. All right. Otherwise, folks, have a good rest of your evening. And I will speak to you all tomorrow.